Thanks very much for that, Matt. Okay, so look, I'd like to thank the organisers for inviting me to speak today. It's a real pleasure to be able to present some of our recent work. I was able to see before I went onto the full screen some very familiar faces in the audience of colleagues and collaborators, so I'm very excited to be able to speak to you as well. So I'm an adult gastroenterologist based in Melbourne. I spend 80% of my time involved in celiac disease research at the Walter and Eliza Hall Institute. And by way of disclosure, I am a consultant for a biotech company called Jason Tye Din, which is developing an immunotherapy for celiac disease. Because I'm kicking off the session this morning, I thought I'd start off with a few general slides. Oh, I forgot to mention that I've been in the field for about 15 years now, which is not very long compared to a lot of the very experienced attendees in the audience today. But I've been talking to doctors about the importance of not missing a diagnosis of celiac disease. And this picture here is actually my wife, who was diagnosed with celiac disease a year and a bit ago. And this is a picture of her with her probably last gluten-containing bread roll. So she's looking quite happy there, but I'm hoping that with more research and discoveries that maybe one day she'll be able to eat bread again, or at least better tasting gluten-free bread. So this first slide really is to just highlight the fact that celiac disease is now appreciated as an autoimmune-like disease driven by dietary gluten from wheat, rye, and barley. We know there's a critical role for certain predisposing genes, particularly the HLA genes, and then there's a role for environment in terms of triggering the onset of disease. We still diagnose it based on typical changes in the small bowel, but there are circulating autoantibodies such as transglutaminase antibodies, and clinical effects include elevated mortality, increased morbidity, and reduced quality of life. Now, I'm showing this slide here by way of introducing the key role of the gluten-specific T cell, but you will hear more about the pathogenesis of celiac disease in the next session from Professor London, so I won't go into it in great detail, but I wanted to draw your attention to the gluten-specific T cell, which is really a fundamental player in the development of celiac disease. It orchestrates the inflammatory response that ultimately leads to the villus atrophy and the inflamed lesion. And it was actually Professor London who was the first to show these gluten-specific T cells in the intestine of people with celiac disease. And my colleague and past PhD supervisor, Bob Anderson, then later on showed another approach which involved people with celiac disease eating bread for 3 days. And then he showed that these gluten-specific T cells could be encouraged to be induced in the bloodstream, and then you could study them in the blood. And several groups have put forward the notion that if you can detect these gluten-specific T cells, this might provide a novel diagnostic approach to celiac disease. And several groups have now mapped the relevant peptides that drive these gluten-specific T cells from wheat, rye, and barley. And some of my work involving the gluten challenge has led to the definition of 3 key peptides within wheat, rye, and barley that are the basis for the immunotherapy of NextVax. I wanted to draw your attention to the gluten-specific T cell because it's going to become relevant later when we talk about oats. So why should we care about oats? Clearly, they're a very desirable grain to add to what is otherwise a very restricted gluten-free diet. And there still remains quite a bit of lingering uncertainty about its safety for at least a proportion of people with celiac disease. And I think we're now in a stage where we have a much better understanding of the immunology of oats and avenine, T cell responses to avenine, and also have the support of serial science technology so that we can now meld an understanding of the immunology with clinical outcomes. And particularly here in Australia and as well as New Zealand, we have very restrictive gluten-free labelling laws. So many of you will be familiar with the fact that we don't have a Codex 20 parts per million limit. It's a no detectable gluten limit here in Australia, which means that our limit is around 3 parts per million or whatever the current ELISA can detect. But also oats are excluded from a gluten-free diet here in Australia. And there's a huge groundswell of support amongst patients who really want to eat oats for research to really try to clarify the safety position. Now, in clinical practice, we often will allow our patients to eat oats as long as they have a baseline gastroscopy, 3-month oat challenge, and a follow-up gastroscopy that shows no damage, and then we may allow them to eat oats. But it's quite a cumbersome procedure, and as you could imagine, it's liable to fail if the person accidentally eats a bit of bread or something before the final gastroscopy and mucks up the results. So there needs to be a better way to identify people who may be at risk of oats toxicity. So this slide really just highlights the benefits of oats in the gluten-free diet. It is a very nutritious cereal packed with a good mix of amino acids. One of its greatest benefits is the presence of beta-glucan, which is a resistant starch that adds viscosity to the meal and seems to reduce postprandial glucose increase in the blood. It also seems to have effects on reducing LDL cholesterol. And I think as emerging evidence now tells us that a long-term strict gluten-free diet, if not done properly, can be associated with higher rates of cardiovascular disease and metabolic syndrome and obesity. I think grains with these kinds of favorable properties are really beneficial if we can include them in the diet of people with celiac disease, if it can be done safely. So this slide really highlights a phylogenetic tree of oats. And you can see here that wheat gluten and the related prolammin proteins that are toxic to people with celiac disease from barley, so that's the hordeins, and also the rye secalins, are very closely related. But the oats sort of diverges off at a different family, but they're still kind of cousins. So there is a relationship there. And there are gluten-like proteins within oats called avenins. And when you look at the structure of avenins, many of the sequences are very similar to those found within wheat, rye, and barley. One important point to note, though, is the actual percentage of avenin within oats is a lot lower. So as you can see on this slide now, if patients consume 100 grams of wheat, barley, or rye, they're getting at least 4 to 11 grams of the relevant toxic prolammin. Whereas if they have 100 grams of oats, and if we know that about 30 gram serve is a standard serve of oats, so 100 grams is a very large amount. That's a very big bowl of oats porridge. That only generates 1 gram of avenin. So automatically we can see that we're operating at a much lower level of potential prolammin toxicity just because of the differences in protein content. Now, the early work of Willem Dijker and his Dutch colleagues back in the 1950s really raised the notion that oats may be unsafe for people with celiac disease, although there were subsequent studies that kind of contradicted that. And I think if we look back at a lot of the earlier studies, I think it's important to be mindful that many didn't really control for wheat contamination in oats. And we know that even a small amount of wheat can cause substantial contamination in the oat supply. And for most commercial brands of oats available in the US and Australia and probably the UK are likely to be contaminated with significant amounts of wheat. So really, if people are going to be eating oats, they need to be dedicated wheat contamination-free oats. And that requires substantial facilities with processing and milling and everything kept separate from contaminant sources like wheat. Now, this here is a busy slide, but it really shows a table that I've lifted from a large systematic review that was published in Gastroenterology last year. And it was a study that looked at all of the kind of clinical studies assessing the safety of oats in the gluten-free diet. And they looked at more than 600 studies and whittled it down to 28. And you can see here on this particular slide, the top 6 studies are randomized controlled trials. So as you would know, the kind of level of evidence which we feel a lot most comfortable about in terms of providing data to inform clinical guidelines. And of those 6 studies, all of them were done in Europe. They all use pure, uncontaminated sources of oats. 3 were in adults, 3 were in children. And you can see across on the right that the readouts were variable. Some looked at GI symptoms. Some also assessed celiac serology or intestinal histology. some also measured intestinal epithelial, intraepithelial lymphocytes as a marker of gut inflammation, and some assessed dermatitis herpetiformis. And if you see green, then there was no change or worsening with oats. If you see yellow there, then it suggests that there was at least some deterioration in a subset of participants, and red means that there was a significant deterioration. And it's clear that the board is pretty much mostly green, so very reassuring that both at a symptomatic, serologic, histologic, and inflammatory level, there's minimal in the way of danger with oats. But you can see that the doses of oats used were variable. There was only a few studies that had large amounts of about 100 grams of oats a day. And of the studies down below, the observational studies all had quite different designs. So different outcomes. And since that's been published, there's been a few more studies that have come up in the literature. A Finnish study that showed that about 82% of Finns with celiac disease are consuming oats. And this was a 10-year follow-up study. So it is a very long period, which is provides some very reassuring data that there was no adverse clinical outcomes in those groups of celiacs consuming oats compared to those who were not consuming oats. And if anything, there was some increased improved quality of life in those celiacs consuming oats. The Italian study was a double-blind, placebo-controlled randomized study. Again, quite decent numbers of children involved. It was a crossover design. They didn't look at histology, but they did show that there was no changes in symptoms, serology, or intestinal permeability. So again, very reassuring. However, not all studies have been entirely reassuring. So a study by Knut Lunden, which was published in Gut in 2003, assessed 19 adults with celiac disease, and they had 50 grams of oats daily for 3 months. One patient definitely developed villus atrophy and a rash. This person improved on an oats-free, gluten-free diet and relapsed quite substantially when she was re-challenged with oats. And about 5 out of the 19 patients had some markers of increased inflammatory cytokines. And you can see on the rest below that there have been other studies that have shown inflammatory changes, both with the IELs or just inflammatory cytokines. So the general sense from this systematic review was that oats are generally safe. However, they rated these randomized controlled trials, the quality as being very low based on attrition bias. So this was dropout in the treatment group. That meant there was an imbalance between them and the control group, or some imprecision in the readouts. And they also noted that several of the studies showed worsening of symptoms after oats intake. And overall, their criticisms related to small sample sizes, or the fact that most studies were done in Europe and the results could not be extrapolated outside of Europe. There was also unclear assessment of compliance to a gluten-free diet in many cases. There's also it's unclear whether the symptoms that people experienced were related to Avenant itself, or whether this was an effect of the fiber load. As you all know, the gluten-free diet is low in fiber. So when you have oats, this is a substantial fiber source. So this could be another source of symptoms. And importantly, most of these studies did not talk about the cultivar of oats. And there's a body of literature now really emerging, suggesting that different cultivars of oats may have different levels of immunotoxicity. And so there was the criticism that we need to be more explicit about which type of oats is being tested. Obviously, study withdrawals may lead to an underestimation of the true adverse impact of oats. And certainly in our experience with all our oats feeding studies, we tend to get a very biased population of people who really like to eat oats and enjoy oats. And those people who feel it makes them unwell tend not to take part in these types of oat feeding studies. So I think this highlights the importance of quality randomized controlled studies, as opposed to just relying on observational studies. And in fact, this was indeed the conclusion by the group that did the systematic review, published just last year, that more rigorous double-blind placebo-controlled studies are needed with oats sourced from different regions. And also another review that was more Europe-centric, with a number of key UK authors present. This is the second publication I've listed there. Also noted that the issue is, I suppose, far from concluded in terms of safety of oats. And there did need to be further work looking at safety of different oat cultivars and identifying people with oats intolerance. So I guess the question is, how can we use our understanding of oats immunology to sort of further the field? And so if we look back at what's gone before, the Dutch group led by Frits Koning published back in 2003 their work that initially predicted several avenine peptides as being potentially immunogenic because of their homology to wheat gluten immunogenic sequences. And they used some T cell lines from the intestine that was raised to these wheat gluten sequences to look at cross-reactivity. And there was some, but it wasn't complete. And then the subsequent year, the Norwegian group led by Ludvig Solid and Knut Lunden then managed to pull out intestinal T cells from people with celiac disease that did respond to these oat avenine peptides. And they were indeed the ones that were predicted by the Dutch group. And this was, I think, they looked at 9 people and managed to pull out at least 4 different T cell lines and 1 T cell clone. So several of these patients were clearly oats intolerant that developed villus atrophy with oats ingestion. But interestingly, some of the patients who did not develop oats intolerance and were able to consume oats safely also had T cells reactive to these avenine peptides. So you'll recall that I mentioned my colleague Bob Anderson, who developed the 3-day gluten challenge technique. And I sort of came on board with him in about 2003 and started my PhD then. And so one of my first studies was actually feeding people oats for 3 days to see if we could induce T cells in their bloodstream. So what did that show? Well, I arbitrarily chose 100 grams of dry oats to feed to patients daily for 3 days. This was cooked into a large bowl of porridge that they consumed. And then I collected their blood on day 6. And we showed that 6 out of 73 adult celiac disease participants had significant elevations in T cells to the specific avenine peptides, including the ones that had been reported previously using intestinal T cells. Interestingly, half the participants had symptoms, particularly gastrointestinal symptoms, but there was no correlation with the immune responses that we were reading out. And as you can see here, the sequences between the oats immunogenic sequence and the barley one are very similar, which had been reported before by the Dutch group. But when we looked at them in more detail, the oats sequences were more susceptible to degradation by gut proteases, and they bound less stably to the HLA-DQ2 molecule, which meant that they probably triggered a T cell response less effectively. So we predicted that there was cross-reactivity from barley driving some of this oats immunity, but we wanted to show that that occurred in vivo. So what we actually did was feed people with celiac disease barley and test whether their T cells recognize these oats sequences. So you can see the little figure in the bottom left-hand corner. And when people consume barley, we were able to induce T cells, and you can see within the red circle, the AV1 and the AV4 sequences there. These are oat avenine sequences, and there were strong responses against avenine, but this didn't occur if people with celiac disease consume wheat or rye. So the implication was that barley-specific T cells were driving the avenine immune response. And these barley-specific T cells were common. They were found in the majority of the 13 or so participants in that study. So the feeling was that there's cross-reactivity by barley that may be driving this oats avenine specific T cell response, but oats is inherently a less immunogenic peptide. So it led us to this hypothesis that avenine specific T cells are ubiquitous because we can see them in the majority of people who eat barley. So this oat response that we see in only a small percentage of people is probably not something that's idiosyncratic to them. It's something that is potentially triggerable in all people with celiac disease if we perhaps push the dose of oats high enough. And so this little schematic diagram there tries to encapsulate that concept that the dose of oats may be important in driving avernin-specific T cell responses, and possibly patient factors are also relevant in determining the subset of people who ultimately develop oats toxicity. But we needed to explore this more, and we needed to understand the link between avernin-specific T cells and how they might link in with sensitivity to oats. And the aim was to really compare those people who responded to oats with T cells, we called them the responders, to the group of non-responders in terms of their clinical outcomes. We wanted to also study a safe threshold of oats to determine whether we can get more people to consume oats in a safe fashion. We wanted to study a range of oat cultivars. And we also wanted to tease out why some people develop symptoms, whether this was an oat avernin immune response effect or whether this was more simply a fiber loading effect. So we needed to consider a blinded randomized trial that controlled for the fiber load there. So we had good intentions, but we came unstuck at the start because one of the big problems is that only about 8% of people, 1 in 12 celiacs, will actually mount a T cell response to these oats sequences. So to try to phenotype this group, to understand more of their clinical outcomes, was getting really challenging because we weren't able to trigger enough T cell responses. So we thought, what about if we can extract avernin from the oats and feed avernin to people? Now, this is not entirely a new concept. And in fact, Dr. Moulton from Birmingham Children's Hospital in 1958 published in the Archives of Disease in Childhood this concept, and I've highlighted it in red there. The possibility of using oat protein itself was explored, but they had some difficulties actually with the technical extraction of that, so they weren't able to proceed. But this doctor certainly had the concept that maybe they could improve the quality of their feeding studies by actually using pure oat protein as opposed to oats in general. Now, this concept has also been used very effectively by my colleague Peter Gibson in Melbourne with the gluten sensitivity studies that he's run, and also with Knut Lunden more recently with this paper where they've used purified gluten instead of wheat bread in challenge studies. And you'll hear more about these studies next, but I think it's a very nice way to segregate the effect of the protein as opposed to the whole grain. So what we did is we reached out to a colleague that I'd worked with several years ago, Greg Tanner, who's a cereal scientist who used to work with CSIRO. And here he is standing in a big field of barley, but it's not normal barley. It's called Kabari barley. It's ultra-low gluten barley, which resulted from some work that I did with him earlier in the day, several years ago, where it was a selective cross-breeding of barley so that the actual hordein content is less than 5 parts per million. Now, this barley is now being exported to Germany, where it's being made into the world's first gluten-free beer that fits the German purity laws. So it is actually truly gluten-free barley by the Codex definition, but unfortunately and ironically, it can't be sold in Australia because of our no detectable gluten limit. Because we can detect gluten in this barley, we can't sell it in Australia. But it's a great Aussie sort of export invention. But Greg has had a lot of experience as a cereal scientist, and I brought him on board to see if he can help us work out how to extract avernin. And to cut a long story short, and I know I'm probably running over time, so I don't want to hold up the program for the day. In a nutshell, we've gotten now 400 kilograms, but we started with 200 kilograms of pure wheat-free oats from Western Australia, grown in a wheat-free sort of farm as best as possible. Visual and electronic sorting to exclude any contaminant grains. This was milled in a wheat-free facility in Melbourne. It was then transported to New South Wales, where they had a lot of alcohol, 300 litres or more of alcohol, which was used to extract the avernin. And you can see the sludge there in the middle of the screen. It's spun down, and then in the bottom left of the screen, we can collect the supernatant, and when it's chilled, the avernin actually precipitates out. So you can see that cloudy solution in the right-hand Falcon tube compared to the clear solution in the left one. That's the avernin being precipitated. You spin that down and you're left with this very nice, pure white, fluffy powder, which is pure avernin or close to pure avernin. We got 1.2 kilograms from 200 kilograms, and we're now processing another 200 kilograms to get an extra kilogram. So we can do more feeding studies. And we're running mass spec on this avenine to confirm it contains the relevant peptides. So the aim is to now conduct a study which hopefully addresses some of the issues that have gone before, in terms of, we want to blind this study. We want to use gluten-free rice porridge that everyone has, and then we spike in the avenine, either at 0 or a low dose, which is equivalent, which we're calling low dose, but it's not really low dose. It's about a decent sized serve of oats, 50 grams of oats, or a high dose. So that's 6 grams a day. And that number is really borrowed from the wheat gluten literature, where we know that definitely triggers a response. And that's 600 grams of oats. So obviously much more than a person could actually eat in a day of oats. So this is going to be a way to really test the hypothesis that the induction of T cells will be much more prominent in this group, and that we hope to correlate it with clinical endpoints. And we'll be using symptom diaries, serology, histology, and a range of immune readouts, including tetramers, which will allow us to determine if the circulating frequency of avenine-specific T cells predicts for oat sensitivity. And we've got a range of different cultivars. We have several different oat growers and companies who've provided different sources, and we've also sourced oats from the UK, Europe, and the United States that we'll be testing in vitro studies. So I guess to conclude, the studies generally support the safety of oats for celiac disease. So there's no cause for alarm for our patients, but obviously ongoing clinical follow-up's always important to detect any issues. There may be a minority of people with celiac disease who do have true oat sensitivity, and hopefully studies like our own and others will help resolve this issue. So I'd just like to finish by thanking my team and collaborators, particularly Greg Tanner and Frank Becks, who have been really fantastic, Kim Rintoul, the farmer who produced the oats, and Amy Barry and her team at Manildra. And I'd particularly like to thank Coeliac UK for the funding support that they've provided for our research, which has really made all of this possible. So thank you very much for your attention.
Fantastic. Jason, I hope you can hear me. I just want to say thank you very much for your fantastic lecture and update on oats. I appreciate it's quite late in the night there, and I'm really grateful on behalf of everyone here for you giving up your time and giving that excellent presentation. Just for the purpose of time, I'm going to not restrict any questions if I may, and wish you a good night, and then I'm going to move on to our next speaker if I may. So thank you very much again, Jason.